Hello, and welcome to this Forth Valley and West Lothian RIC supported study resource. My name is Miss McEwen, and today's lesson is going to be focusing on problem solving and numeracy skills in National 5 biology. The learning intention for today's lesson is to develop our understanding of how to answer problem solving questions with particular reference to unit-specific numeracy skills and graphs. In order to achieve this learning intention, we must tick off the following success criteria. To be able to calculate cell sizes in terms of length and breadth, to determine base pair numbers from given information, to be able to calculate cell division rates, to develop skills for identifying information from graphs. Throughout this lesson, you will see various symbols on the slides to indicate what you should do. Here are the symbols which may be used and what they mean. There is a symbol for listen, read, write or copy, correct your work, and pause the video to complete the task. Where there is a write or copy symbol, this is where I would recommend taking a note or diagram. When I asked pause the video, please complete the question that I have set and take a couple of minutes before you play the video. And when you've finished writing your answer for a question, then replay the video, and when you see correct your work, I will go over the answer. For this lesson, you will need your jotter or a piece of paper, a pen or a pencil, and a calculator. The first topic we are going to go over is from Key Area 1, Cell Structure in Unit 1. We're going to focus on cell size. As backup information, all living things are made from cells. Cells cannot usually be viewed with the naked eye. You must use a microscope to view cells and to see the detail and structures which make up cells. This slide is a read-only slide, but feel free to take any additional notes you wish throughout the PowerPoint. There are subject-specific numeracy skills in this topic. You may be asked to calculate a cell size. Here's our first example. The diagram below shows a group of cells as seen under a microscope. The field of view was 2 mm in diameter. Calculate the average length and width of the cells. Luckily, this example has a diagram. If there is a diagram, please use it as it will have key information to help you solve the question. My first recommendation when doing any biology question in an assessment or SQA past paper is to look for information in the diagram. We first want to calculate the length, which I have labelled here. To calculate the length, I need to count the number of cells from the top to the bottom of this microscope slide. If you have a look at the highlighted area, there are 10 cells going from the top to the bottom of this circle. I then look to see if the question has provided any additional information, which luckily it has, giving us a 2 mm diameter in total. To calculate the average length of one cell, I would do a division here. I would divide 10 cells by 2 mm in diameter, which gives you 0.2 mm per cell. This is the answer to the first half of your question. Next, we need to calculate the width. So we want to do the same thing again, but this time we want to count the number of cells going from left to right. If you count along using the highlighted section here, there are 5 cells. This time we want to do another division, but using this number. Calculating an average width, we would then divide 5 cells divided by 2 millimeters in diameter, which equals 0.4 millimeters per cell. So the average length of a cell in this example is 0.2 millimeters, and the average width of a cell is 0.4 millimeters. Example 2. The field of view of a light microscope measures 2 millimeters in diameter. 20 plant cells were counted in a line across the diameter. 1 millimeter equals 1000 micrometers. Calculate the average size of a cell in micrometers. This example deliberately does not have a diagram, which means you have to imagine in your head what those cells would look like counted across the diameter, or you can draw a diagram on your SQA past paper or in your jotter to help you with this, if visuals help you learn. What I would recommend doing to solve this question, the first thing is to look to the question for any key pieces of information. Here we've seen the diameter is 2 millimeters, and there were 20 plant cells counted in a line across the diameter. So now I know I can do my division. So to calculate the average size of a cell, I divide 20 by 2, so I divide the 20 cells by 2 millimeters in diameter, and I get 0.1 millimeters per cell. Now unfortunately we're not finished here. This example is slightly more difficult. The second thing to notice from the question is any conversions they provide you. They have provided us here with a conversion of 1 millimeter, which equals 1000 micrometers. If you ever see any conversions like this, we must use it. It's very important in order to calculate the correct answer. If you notice, our answer at the moment is in millimeters, but the answer to the question they would like in micrometers. So we're not yet finished. If you were to place 0.1 as your answer here, unfortunately you would get this wrong. So what do we do next? We must convert millimeters to micrometers. If you've never seen the sign before for the units of micrometers, it is a different shaped UM. So if 1 millimeter equals 1000 micrometers, provided from the question, then I must times my answer or multiply it by 1000. So 0.1 millimeters becomes 100 micrometers, as 0.1 times 1000 is 100. Now my answer is in micrometers, I can write this answer in. So we now know the average size of a cell is 100 micrometers. And I have a question for you to try. You will notice the pause this video sign in the bottom right hand corner. The diagram below represents plant cells on a microscope slide. 1 millimeter equals 1000 micrometers. Which is the length of an average plant cell in micrometers? A, 320, B, 340, C, 360, or D, 380. Numeracy style questions for cell size may come up in both sections of the past paper. You may find them in the multiple choice section of the past paper, like this question here, or you may find them in section 2. They will usually be one of the first questions in whatever section it is found in. Please pause the video here to try this question out using the hints and tips I provided in the previous 2 examples. When you're ready, press play and we will go over your answer. We're now ready to correct your answer. The answer was B, 340. How did we get to this answer? The first thing I always recommend doing is looking to the question for key information. Here I've highlighted that the diameter of the microscope slide is 1.7 millimeters. I've also seen a conversion. 1 millimeter equals 1000 micrometers. These are both key and essential pieces of information for you to calculate the average length of the plant cell. I then count along because I've got the additional diagram which I must use, and I've noticed that there are 5 cells in the length going across the diameter. So the first thing I do is my division. I divide 5 by 1.7 millimeters, which gives me an answer of 0.34 millimeters. Again, be careful to check your units here, as our answer is in millimeters, but the answer to the question they would like in micrometers. So now we must use the conversion. We must now convert millimeters to micrometers, and if 1 millimeter equals 1000 micrometers, then 0.34 millimeters equals 340 micrometers. Then we can see that 340 matches up to answer B. The next key area we're going to look at is key area 4 of Unit 1, which is DNA and proteins. We're going to narrow in on DNA base pairing in particular. For background information, DNA is also known as deoxyribonucleic acid and is stored in the nucleus of every cell. It is a code made up of 4 letters: A, T, C, and G. At National 5 level, you are able to just write the letters, but always make sure these are in capitals. For more information, A stands for adenine, T stands for thymine, C stands for cytosine, and G stands for guanine. DNA is a double-stranded helix, which looks like a twisted ladder or staircase. The 2 strands are held together by complementary base pairing between the bases. What this means is A will always bind with T and T with A, and C will always bind with G and G with C. This is key to answering the numeracy skills based on DNA. Again, this was a read-only slide, but feel free to take down any additional information that you will find helpful. Our first example of these questions is here in example 1. A single strand of DNA contains 830 adenine, 929 cytosine, 774 guanine, and 615 thymine bases. How many guanine bases would be in the complementary strand? A, 615, B, 774, C, 830, or D, 929. The first thing to notice here is that the example of answers all match exactly the numbers of bases which have been provided in the question. This is very important as we must remember our base pairing rules and which particular base matches with its complementary base. To go about answering these questions, again, my first recommendation is to look for key information in the question. I would either highlight these or underline. Here I have highlighted 850 adenine bases, 929 cytosine, 774 guanine, and 615 thymine bases on a single strand. But the question is asking us for guanine bases in the complementary strand. To answer these questions, we must first remember our base pairing rules. So A and T's always bind together, and C and G's always bind together. How I answer these questions, and you may find it helpful too, is to put down some working. I write down all of my bases, A, T, C and G, down the left-hand side of my page, and I then write down any numbers of these bases that I can find from the question. And luckily, I know the exact amount of each base on that one single strand, so I can fill that in. But the question is asking to find out how many guanine bases there will be. So next what I do is for the complementary strand is I write down the letters down the right hand side. Now notice these are not the same as the left because I must use the base pairing rules. So I know that A binds with T, T binds with A, C binds with G, and G binds with C. If I have 830 bases on the first single strand, then the left hand side, because that binds with T, I must also have exactly 830 thymine bases on the complementary strand, which is down the right hand side of my working. So if we want to figure out how many guanine bases there would be, we need the exact same number as the cytosine bases on the first strand, which was 929. Again, we double check our answers and we can see the answer would be D, 929. Now I have two questions for you to try. The first one is an easier example and the second one is slightly more difficult, so you will need to think about number 2. Please pause the video here and try these two questions. Give yourself about 5 or 6 minutes for this. When you're ready, press play and we will correct your answers. Question 1. A DNA molecule consists of 3,600 bases. 220 bases are thymine on one strand. Calculate the number of adenine bases on the complementary strand. First thing I do, as usual, was to highlight any key information from the question. I must also remember our base pairing rules, A binding with T and C binding with G. This may be beneficial to note down at the side of your paper. A to T and C to G. Again, I would list the bases down the left-hand side of one strand and any information I know. So from the question, I know that there are 220 bases which are thymine on the one strand. This helps me then calculate how many adenine bases are on the additional strand, which is complementary. If it's complementary, that means if there are 220 thymines on one strand, there must be 220 adenine bases on the complementary strand, because A and T's pair together. They are complementary base pairs. Which means our answer to this question was 220. 220 adenine bases. Question 2. In a DNA molecule made up of 760 bases, 114 bases are cytosine. Calculate the percentage of adenine bases in this molecule of DNA. So here we're looking at the one molecule of DNA in total. Again, this is slightly more difficult and will make you do further calculations. The first thing we do is highlight any key information in the question. So we know there are 760 bases in total, and 114 bases of these are cytosine. But we want to work out the percentage of adenine. So let's write down some working. First thing to always remember is the base pairing rules. A will always bind with T and vice versa. C will always bind with G and vice versa. I would then write down the 4 bases on the one strand. A, T, C and G is on the left hand side of my paper. From the question, I know there are 114 bases that are cytosine, so I can fill in this answer next to C. What can I do next with only this amount of information? Well, if I remember my base pairing rules, I know that C always binds with G. So in total on this molecule of DNA, if I have 114 cytosine bases, I must have 114 guanine bases, as C and G's always pair together, so we should have even numbers. Next, what I can do is add these two numbers together, giving me a total of 228 bases, which are C and G. The reason I add them together and figure out this number is because next, if I know there are 760 bases in total, and I know that 228 of these are cytosine and guanine, I can take this number away and do a subtraction to get 532. So 760 bases in total, take away the 228 bases which were made up of C and G, gives me 532 bases left over. I now know that 532 bases out of the 760 are adenine and thymine, but the question only wants adenine. So all I do with this number is half it. I divide 532 by 2, or half it, to get 266, which means I have 266 thymines and 266 adenines. Unfortunately, this question is not done yet, as if we read the question over again, we are looking for the percentage of adenine bases, not the number. So 266 is just how many adenine bases we have. We want to know the percentage of adenine bases out of the whole molecule of DNA. So we must now calculate a percentage. To do that, we would divide the 266 adenine bases divided by 760 bases in total and times this by 100, giving us an answer of 35%. We now know there are 35% of the bases in a molecule of DNA which are adenine. The next key area we'll look at is mitosis and cell division, which is usually found in Unit 2. Again, there are numeracy style questions within this key area. For background information, most animals and plants start off life as just a single cell, but grow to become adults containing billions and billions of cells. The type of cell division that makes animals and plants grow is called mitosis. In mitosis, a parent cell divides into two identical daughter cells. These daughter cells divide in two, and so on and so on. Again, this is a read-only slide, but feel free to take any information and/or diagrams if you feel they will help. The diagram here, we have, to explain it, we have one parent cell which divides into two identical daughter cells. Each of these cells would then go on and divide into two more identical daughter cells, and so on and so on. So if we have a situation where a cell divides every 30 minutes, how many cells will there be after 1 hour? This is where the numeracy comes into play. If I have one cell, which should always start off at 0 minutes because you need one cell to be there to divide. I know these cells divide every 30 minutes, and I know one cell divides into two. So at 30 minutes, we have two cells. Each of these cells will then divide again and again, each cell dividing into a further two. So after a cell divides every 30 minutes, after 1 hour, you will have 4 cells. How to approach these questions? The best way to go about answering these types of questions is to figure out how often the cell divides. For example, these cells divide every 20 minutes. From there, we want to figure out your end time. Look at the question to figure this out. So for example, again, how many cells will there be after 100 minutes? Now you can either draw out the division diagram that I've previously showed you, but that can get slightly muddled when you're dealing with big numbers of cells. You could also write it out to see how many there are. I'm going to show you another way which I think is clearer and more concise to help you get to the answer. This is by using a simple t-table. I fill in the headings of my t-table with minutes and cells, And that's because we are trying to work out how many minutes it will take or how many sales there will be in 100 minutes. It doesn't matter which way round you put these headings. So if you put sales on the left hand side and minutes on the right hand side, that's totally fine as well. Because I know from the question that the sale divides every 20 minutes, I can then go up in periods of 20 minutes from 0, 20, 40, 60, 80 and 100. And the reason I stop at 100 minutes is because, again, I've looked for clues in the question and the question states how many sales will there be after 100 minutes. So we must stop at 100 minutes. I know that we always need to start off with one cell at 0 minutes, and I know that one cell divides into 2. So every 20 minutes a cell divides. So after 20 minutes, one cell would become 2 cells. After a further 20 minutes, these 2 cells would undergo divisions and become 4 cells. After another division, 8 cells. Again, there would be 16, and again there would be 32. Notice that we double the answers every time. I then stop at 100 minutes, as that's the answer to the question, so I know there are 32 cells after 100 minutes. A key point here, the trick is to remember to double your cells and not add 2. So we make sure that we go up 1, 2, 4, 8, 16, 32. These will always be the numbers that you will go down the list or the T table, and they shouldn't change in any way because you're always doubling your answer and not adding 2. So that is a helpful hint to remember. Here's a question for you. Example 1. A cell divides every 5 minutes. How many cells will be present after 1 hour? A 32, B 128, C 64 or D 12? Please pause the video here and try this question now. Take a couple of minutes to do so, and when you have an answer, press play and we will correct your answer. We are now ready to correct your answer. So if a cell divides every 5 minutes, how many cells will be present after 30 minutes? I draw my T table, I add my headings, minutes and cells, and this time I know that the cells divide every 5 minutes, so I can go up in periods of 5 minutes. I know where to stop because the question has told me how many cells will be present after 30 minutes. So I go up in 5 minutes, 10, 15, 20, 25, and I stop at 30. On the other side, I would start off with one cell and I would double my answer after every 5 minutes. I would have 1, 2, 4, 8, 16, 32, and 64 cells. I know to stop here because the question has told me to find out how many cells will be present after 30 minutes. To check our answer is correct, because this is multiple choice, it must be one of the 4 options. We have 64, and if you have a look at our options, you will see our answer would be C. Here is another example, example 2. This time, and it's worth noting here, these questions can be asked in terms of mitosis, or they can also be asked in terms of bacterial cells, so you may find them with questions in relation to cell structure. The bacteria have an initial concentration of 1000 cells per centimetres cubed. Each cell divides once every 30 minutes. Calculate how long it will take for the concentration to become greater than 15,000 cells per centimetres cubed. Again, pause the video here, using any tips and tricks that I have taught to you previously in the other 2 examples to try and solve this question. Again, take a couple of minutes. When you think you have an answer, press play and we will correct your answer. We're now ready to correct your answer. Okay, what do we do first? We draw our T-table, we add our headings. The next thing we do is have a look to the question for key pieces of information. The first thing we come across is the initial concentration of 1,000 cells per centimetre cubed, which means we can add this in under our cells column. We would usually start with one cell here, but the question has specifically told us we have an initial concentration, which means at the start we have 1,000 cells. Next, we notice some more key information. We now know that the cell divides every 30 minutes, and we are looking for how long it will take for the concentration to become greater than 15,000 cells. So this time we are not looking for the number of cells, this time we are looking for the time. I can double my cells every time because I know after each division, remember, one cell divides into two and so on. So we double our answers. So we would have 1,000 going to 2,000, going to 4,000, going to 8,000, and you would keep going until you reach a number which is higher than 15,000. The first answer we get to is 16,000. So I know to stop here. On the other side, I know that we divide every 30 minutes. So I've went up in 0, 30, 60, 90 and 120 minutes. Again, I know to stop here because we have reached a concentration greater than 15,000 cells as stated in the question. My answer for this question should be 120 minutes, or you may have written 2 hours. Both of these would be acceptable. The last type of questions we're going to look at are the skills when interpreting graphs. These are general numeracy skills and will be throughout your past paper and could be relevant to any unit or key area. You will always have at least one graph and probably more in any SQA past paper. The graph below shows the size of a human's brain and ovaries from birth to maturity as a percentage of final size. Compare the growth of the brain during the first 4 years with growth from age 16 to 20. And we have 2 questions. Let's look at question 1 first. At what age did ovaries reach 40% of their final size? The first thing I do here, or recommend doing here, is looking for key information from the question. First of all, I am looking for the ovaries, and second of all, I want to see at what age did they reach 40% of their final size. The next thing I would recommend doing is either highlighting or making sure you are looking at the correct trend in the graph. This graph is showing the age in years from 0 to 20 along the x-axis and the size as percentage of final size from 0 to 120 along the y-axis. We then have a solid black line showing the development of the brain, and we have a dotted line showing the development of the ovaries. We need to ensure that we are only looking at the dotted line for the trend of development of ovaries, as this is what the question is asking us. I want to see the final size of the ovaries in order to find out or calculate 40% of this final size. So I look at the last plot on this trend, and it is here at age 20, and I follow across to the y-axis to find out that it is 100. I have used arrows here to highlight this on the graph. Now that I know that the final size of the ovaries was 100 as a percentage of the final size, at age 20, I can calculate 40% of this. So in order to do this, I must calculate 40% of 100. Therefore, 40 divided by 100 times 100 equals 40, as I'm working out a percentage. Now with this number, I can look back to the graph, again ensuring that I'm only looking at the dotted line to see when or what age the ovaries were 40 as a size as percentage of the final size. I look at 40 and I go across the graph using the arrow that I've used to highlight, and I find a plot just above age 16. So my answer to this question, at what age did ovaries reach 40% of their final size? The answer is age 16. Question 2. During which 2-year period was there greatest growth of the ovaries? Again, I want to make sure I am only looking at the dotted line for the trend of development of the ovaries to answer this question. I underline or highlight key information to help me solve the answer. Now I know I'm looking for a 2-year period with the greatest growth of ovaries. To answer this question, we must identify the largest difference in size of the ovaries over a 2-year period by looking at the graph. This is a selecting question, which means the answers are in the graph, but we must select the correct answer. We must look along the dotted line for the trend and development of the ovaries, and we must look for the biggest difference in a 2-year period. Note that I have highlighted the biggest difference here, which is between 16 years and 18 years. This would be our answer. The reason this is the answer is because this is a difference of 40. At 16 years, we were at 40 as a size of percentage of final size, and at 18 years, the plot was at 80. Next is a question for you to try. The graph below shows changes in the enzyme and substrate concentrations in a seed over a period of time. How many days does it take for the substrate concentration to decrease by 50%? A 2, B 3, C 4, or D 5? The first thing to notice is that this is an A-level question. The easiest way to notice A-level questions in an exam paper or when practicing questions is to notice that they have an additional scale down the right-hand side. Another thing is to notice if they have 2 or more trends to follow on the graph. The trends are clearly labelled on the left and right-hand sides of this diagram. Enzyme concentration is the solid black line, and substrate concentration is the dotted line. We have time and days along the x-axis. We have enzyme concentration in units on the left-hand side, and we have substrate concentration in units on the right-hand side. Please pause the video here to try this question. Give yourself 2 to 3 minutes. When you have an answer, press play on the video and we will correct it to see how you got on. We are now ready to correct your answer. So, the first thing I would do here, or recommend doing, is again looking at the question for any key information I can use to help solve the question. Here I've underlined or highlighted, we are looking at the substrate concentration to decrease by 50%. When dealing with an A-level question, the first thing you should do is to ensure we are using the correct scale and looking at the correct trend to find our answers. This is where some people can trip up by looking at the wrong scale and writing down the wrong answer, unfortunately. So here I've highlighted from information in the question that we know we're looking at substrate concentration. So straight away, I know that we're looking at the dotted line. And because we're looking at the dotted line, we have to use the additional scale. So we are looking at the right-hand side scale, 0, 1, 2, 3, 4, 5, 6. This may be helpful for you to do in an exam, either highlighting the correct scale or highlighting the correct trend or line on the graph so that you can easily follow this. Now we need to identify the highest substrate concentration because we need to do this first so that we can then decrease it by 50%. The highest substrate concentration comes from the highest plotted point on the graph by looking at the dotted line. If you have a look, the highest point on the dotted line is 6. Be careful, we must look at the right-hand side of the scale as this is the substrate concentration in units, If we were to write 14 by looking at the left-hand side, our answer would be wrong because that would be the enzyme concentration in units. So although we're looking at the dotted line, we must always follow these plots along to the right-hand side. We then calculate 50% of 6, which is 3. So 6 divided by 2 is 3. We then go to 3 on the substrate concentration of the graph and find the point on this line. So we follow the dotted line down until the plot would hit 3 on the graph. This is exactly 4 days. We look back at our answers and we can see the answer would be C, 4. We have now come to the end of the lesson and are celebrating success. The learning intention was to develop our understanding of how to answer problem-solving questions with particular reference to unit-specific numeracy skills and graphs. We had four success criteria to achieve. You can now calculate cell sizes in terms of length and breadth, determine base pair numbers from given information, calculate cell division rates, and identify and select information from graphs. Next steps. I would recommend continuing your studying by practicing more numeracy style questions. This will only increase your confidence if these questions were to come up in an upcoming assessment, past paper, or exam. You can access National 5 Biology past papers and their marking instructions on the SQA website. Just type in SQA National 5 past papers into your search bar and the first result should take you directly to this page. There will be up to 4 years of past papers found here to practice more numeracy style questions. The more and more you practice these problem solving skills and numeracy style questions, the better your confidence will increase when answering these, and you will be more ready to answer and solve these questions in any upcoming assessments. Thank you, and I hope you have found this video helpful.